Moving on to chapter 4. The last couple of lectures we talked about single genes in the first lecture, and then in the second lecture, independent assortment and inheritance of multiple genes. This chapter we're actually going to talk about if you were to inherit genes that were on the same chromosome. So this is ignoring the rule of independent assortment. So we're getting away from Mendel's ratios, and we're going to talk about genes that are on the same chromosome, physically attached to one another, and kind of the rules of inheritance and actually being able to map the distance in between these two genes. Once again, here's the chapter 4 outline for your reference, some of the things that we're going to talk about. Some of these things, like chi-square, we're actually going to do in the class, not online. So some of these things may be absent, but they are in your textbook for that, for your reference as well. So here we have a recombination or linkage-based map of some traits seen in Drosophila. The Drosophila, remember, these are fruit flies. You'll have them in your kitchen, little tiny flies. So these are the traits here are traits that are represented or expressed by a single gene. So they're phenotypes that occur from a single gene. And through this recombination mapping, we can actually position them on a chromosome to know how far apart they are. These units on the left-hand side here, these are what's called recombination distances. Sometimes they're denoted as centimorgans, and sometimes they're denoted as recombination distance. We'll get in how to calculate these in just a second. So one of the analogies that the book makes is talking about different maps. And so in the book, what it talks about is having a city map or a street level map and a subway map. So if you're trying to get around a town, having a subway map to show what way you can travel fastest from one side of the town to another, on top of having a street map with, post office and all these areas of interest mapped on it, combining the two gives you really good knowledge of what's going on. And so with genetics, we have what's called linkage mapping, which is knowing the relative position of genes one to another. So gene A is between gene B and C, for instance. And then on top of that, we can have physical mapping. We can actually know the sequence of the gene and then where it physically maps onto the chromosome within the context of the other sequences of the chromosome. So with linked genes, as opposed to independently assorting genes, linked genes or linked alleles tend to be inherited together. So if you're physically on the same chromosome, the odds of you being passed down together is much higher than if you were to be separated because you're physically connected to one another. Now, it's not impossible. We'll get into crossing over and some of these other things that can break up these linked alleles, but for the most part, your highest odds if you are on the same chromosome is that you're going to be passed down together. So in this example here, we see that we have a cross between these two parents, the gold parent here on the left, and then the brown parent here on the right. We have two genes, PR and VG. So one parent is PR-VG, the other is PR plus VG plus. The gametes produced from these individuals are just like the parental type, PR and VG for the gold parent, and then PR plus VG plus for the brown parent. And then the F1, the first filial generation, has one of the gold parental type chromosomes and one of the brown parental type chromosomes. So these are PR plus and VG plus are inherited together, and PR and VG are inherited together because they're physically attached to one another. So they're not going to get mixed up at this point. Now, in some cases, it's possible that these two genes on the same chromosome do get mixed up and kind of swapped for one another. And this process is called crossing over or recombination. So in this example here, we have the two chromosomes lining up. We have the PR and VG from the brown parent on the top here, and then we have PR plus and VG plus from the gold parent on the bottom. And when they line up together going through this process of meiosis, these chromosomes can actually kind of mash together and connect in the middle and swap ends. So what happens is, instead of having a brown and a gold chromosome come out the other side, you've actually recombined these alleles. So you have two chromosomes that are half brown and half gold. And so we have a new combination of alleles here where we started with PR and VG and then the 2+ alleles separately on two different chromosomes. Now both of these chromosomes have 1+ allele and one of the regular PR or VG. Okay, so here's a microscope image of this process actually happening. So I'm going to trace these chromosomes here. We have chromosome 1 on the left here, and then we have the green chromosome here on the right-hand side. And those arrows indicate the point where these two chromosomes are interacting and crossing over is going to occur. So eventually we'll have between these three points, there can be swapping of the chromosomes, and we can get a remixture of the alleles that were previously linked on each of these chromosomes. So the way this was discovered was in an experiment with Drosophila. There was two chromosomes that were in this F1 individual. One had a very dark staining region of DNA, so when they added this DNA stain, this knob here on the right-hand side would stain very dark, and it was only present in one of the two chromosomes. In addition, there was an extra long tail of DNA on this chromosome that also had that dark staining knob. And this unique chromosome that had the long tail and the dark staining region had the two dominant forms of this WX and the C allele. On the other chromosome, we had a recessive WX and we had a recessive C, and it did not have the knob or the long tail. Well, after meiosis in the offspring of this F1 individual, they examined these two chromosomes again, and they found that they now had a recessive WX partnered with a dominant C, which was not in one of those two parental genotypes, and also this knob was inherited along with this dominant C. The other chromosome had the dominant WX and the recessive C, but it inherited this long tail. So what must have happened was these two chromosomes were cut right down the middle and then pasted to one another through this process of recombination. It's important to note that crossing over happens between chromatids and not chromosomes. That is to say that the crossing over happens during the second set of the prophase, metaphase, anaphase, telophase of meiosis and not early on in the process. And so if crossing over were to happen early on, when there's just two chromosomes, as in the top case here, we would end up with two identical sets of daughter cells. So we'd have a pair that are dominant A, recessive B, and a pair that are recessive A, dominant B. However, when we actually look at the outcome of crossing over, we see that we have four individual sets of, or individuals that are not identical to one another in the daughter cells, right? So we have dominant A, dominant B, dominant A, recessive B, recessive A, dominant B, recessive A, recessive B. And so this is evidence that crossing over actually happens during the second set of cell divisions of meiosis. To make things even more confusing, each chromatid can actually participate in more than one crossover event. So here we're at the 4-chromatid stage. So remember, chromatid is a replicated part of a chromosome. So we're at the 4-chromatid stage, and we see that this first chromosome is crossing over, the gold chromosome is crossing over with the first pair of the brown chromosome. The second chromatid of the gold parent is crossing over with the brown, and then with the other half of the gold parent. This third chromatid down is crossing over only with this second gold chromatid, and then the fourth chromatid is crossing over with nobody. So what we end up with, the 4 daughter chromatids of this, this pairing, we end up with 4 completely different mixtures, 1, 2, 3, 4 of those 4 alleles. We'll get more into these multiple crossover events when we actually do recombination mapping in class. One additional note between these multiple crossovers, depending on where the crossovers happen in between which chromatids, we end up with different mixtures of these tetragenotypes at the end of our meiosis. And so if you were to compare these 4 daughter cells to the previous slide, you'll see that all 4 of them have a different mixture of alleles than the last cell, or than the last slide. And that's because these crossovers are in different positions. So depending on how these crossover events occur and what positions between these sister chromatids, we can have a whole myriad of different mixtures in our outcome. So here we have kind of a recap of crossing over, and it's important to remember that recombinants are the result of crossing over between non-sister chromatids. Because if there was a crossover, say right here, between these 2 chromatids, we would have the exact same outcome, right? So if they're homozygous for the dominant or the recessive allele, then recombination would lead to no detectable change in the daughter cells. However, the crossing over, if it were to happen between non-sister chromatids, like is the case down here at the bottom, we end up with 2 of the chromatids that are no longer representing the parental type, and these we would consider recombinants. So now what we're going to look at is how do we determine if 2 genes are linked and they're physically on the same chromosome, or independently assorting, kind of like with Mendel's ratios that we talked about in the last chapter. So similar to the dihybrid cross in the previous chapter, what we're going to do is we're going to set up a cross where we have one parent that is homozygous for both the dominant alleles, A and B, versus a parent that is homozygous for both the recessive alleles, small a and small b. The outcome of that is that F1 generation, the first filial generation, and we have a heterozygote who has a dominant copy of A and B and a recessive copy of A and B. So now in order to determine if this is independent assortment where both genes are on separate chromosomes, or if these are 2 linked genes, we perform a test cross. Remember, like the previous chapter we talked about, a test cross is when you cross this F1 individual to an individual that is homozygous recessive for both alleles. So in this case, small a, small b, small b. So from this test cross, we have 4 possible combination of alleles. Because the tester is only contributing a recessive A, recessive B for both chromosomes, that limits the number of possible outcomes that we can have. So we have a first outcome where we have a homozygous for both A and B, along with the contribution of the tester, the recessive A and the recessive B. We have a individual that is homozygous for the recessive allele here, and also homozygous for the recessive allele from that contribution of the tester. And then we have our 2 recombinants, right? So we have a homozygous dominant for A and recessive for B, and then a recessive for A and dominant for B. These are the products of crossing over events. Now, if these events are independent assortment, then we should expect to see these 4, the progeny of a test cross from 2 genes, to be in a 1:1:1:1 ratio, right? Or 25% for each of the 4 outcomes. Now, if we see that the parental types, the ones that match these 2 parents up here, if they are greater than 1/4, and then the recombinants are less than 1/4, then we assume that they're linked. And one of the ways that we can test this is through chi-squared, where we looked at observed and expected. That is a tool set that will work out in class and not here on the lecture. So one trait that is important to remember with recombination is that if we're looking at 2 genes, the further they are apart, the more likely we are to have a recombination event occur between these 2 genes. So in this picture here, we have 4 different examples of recombination events. Now, in this first example, we have a recombination that occurs between B and C. The second example, we have another recombination event between B and C, but further down the chromosome. In the third example, we have 2 recombination events between B and C. And then in our fourth example here, we have a single recombination event between A and B. Now, because recombination happens randomly across the entire chromosome, the further the distance apart these 2 genes are, the more likely we are to have recombination happen between them. If 2 genes are very close together, they're very closely linked, then the odds are of this random event happening between those 2 genes is very, very small. So because of this, looking at the offspring of this test cross that we looked at in the previous example, if we have a large amount of offspring that show recombination between B and C, we can assume that they have a greater map distance or a greater recombination distance between them than A and B, because there was fewer recombinants between gene A and gene B. So we're going to actually, if you don't understand this right now, that's okay. Just know that the greater the distance between genes, the more likely there is to be a recombination event in between them. We're going to do practice problems of this in class, and we'll actually be able to do the calculations to measure out the distance between them. But the key takeaway, like I said, remember that greater distance means more likely recombination between them. Small distance means less likely recombination. Okay, so this is a pretty important slide that you should definitely look into memorizing these ratios because they're important. The phenotypic ratios from Mendelian genetics and some of these linkage that we talked about are indicators of what type of cross or where these genes are, right? And so we started in chapter 2 with monohybrid crosses, and this is when we're looking at a single gene, dominant and recessive. And for the monohybrid test cross, we saw that all the offspring arose in a ratio of 1 to 1. In the monohybrid selfing, we saw that all of the offspring arise in a 3 to 1 ratio. The dihybrid test cross, which is in chapter 3, where we looked at 2 genes, in the test cross we saw a 1 to 1 to 1 to 1 ratio. And then in the dihybrid selfing, we saw this 9 to 3 to 3 to 1 ratio. Remember, these top 4 categories here, it assumed that IA, independent assortment, right? Now, if we do a dihybrid test cross and we see ratios such as this one, where they're not in a 1 to 1 to 1 to 1 ratio, then we can assume that the genes are linked, right? Because they're not assorting independently. A trihybrid test cross, we didn't really do any examples here, but that's looking at where we have 3 genes and we're assuming they're all on separate chromosomes. And again, the test cross, we'll see this 1 to 1 to 1 to 1 to 1 to 1 to 1 ratio. But in an example where they are not assorting independently, we'll deviate from that 1 to 1 to 1 to 1 to 1 to 1 to 1 ratio, and we'll get ratios that are not equal parts. And so that's because these genes are linked and they don't have that 50-50 shot of being passed down to one gamete or the other. So I'm going to take a brief second here to tell you about molecular markers. Molecular markers are little pieces of DNA that are something unique that we can use to identify an individual with. So you may have heard of single nucleotide polymorphisms. We like to abbreviate that SNPs. SNPs are when you have a gene that has a single change in a single base. So between these 2 individuals here, we see that the GC is changed to an AT. So we would call that a single nucleotide polymorphism, or a SNP. Another type of molecular marker is a mini satellite or a microsatellite. Mini satellites are repeated sequences that are usually 15 to 100 nucleotides long. So I have here an example where we have, right here, the sequence repeated 10 times. So we'd see that's a copy number variant. So we would say this individual has 10 copies of this mini satellite, and the person sitting next to you may have only 5 copies of it, or may have 100 copies of it. And so that's something that we can use to distinguish two people's DNA. Microsatellites are much shorter, and so they're sometimes 2 or 3 base pairs repeating, but they can be up to up to 15 when they become a mini satellite. But these are all unique characteristics of somebody's DNA that we can use to distinguish from one another. So now why this is important is because some of these microsatellites or molecular markers like SNPs or mini satellites as well, we can use these to distinguish individuals who may have a susceptibility to a disease because of that linkage we talked about earlier. Some of these satellites or molecular markers are very, very close to a gene that has a predisposition for disease, say a Parkinson's or Huntington's gene. Well, the copy number variant, because they're so close to those genes, is usually inherited along with that gene because there's not enough room for a combination to happen in between them. So some individuals that have high copy numbers may also inherit that defective gene. So in this case here, we see that we have these individuals that we have the female over here, we have the male over here, and we see that it's kind of hard to tell because they use the letter P, so it's hard to tell the lowercase from the uppercase, but one of these chromosomes here has a dominant allele for this disease, this uppercase P. And so this, because of the linkage, the closeness between these molecular markers, this copy number variant and this gene, there hasn't been much opportunity for a combination to happen in between it. So when we see an individual that has 5 copies of this molecular marker, this copy number microsatellite copy number variant, we can assume that they have also inherited this disease-causing dominant allele. Now, if we look down here at the pedigree, we see that anybody who has the M double prime, which is this right here, when we run that on a PCR, because the copy numbers mean a different length of DNA, so if you have 5 copies, that's obviously longer than 2 copies or 3 copies, but it's much shorter than 10 or 11 copies. They'll all migrate the same distance on this gel, right? And so they create these bands for individuals that have that M double prime, which is the 5 copies of that microsatellite. Now, if we map a pedigree to that of people who have a disease, we also see that everybody in this pedigree who is a dark circle, who has the disease, also has that band that we're looking at, right? So lanes 1, 2, 5, and 6 all have that band of M double prime, and they all have the disease. So what we can do then is assume that this M double prime is a molecular marker for this disease, and we can then go and use that to say, if you have this 5 copy variant of this molecular marker, maybe you should look into if you're going to develop this disease in the future because they're tightly linked and one is indicative of the other. So real briefly, we're going to look at the actual way that this crossing over happens. We're going to get a lot more into this when we get into some later chapters where we look at the molecular machinery involved in double-strand breaks and double-strand break repair, etc. But briefly, the 2 DNAs, the sister chromatids, will have a bubble occur where a double-strand break is happening. And then there's this erosion of the strands where there becomes these 3' overhangs. So 3' being the region of DNA. We'll talk more about that as we get into the molecular methods as well. And one of the strands will then invade this intact double strand and it will form bonds with the complementary bases on that strand. After that strand invades the intact strand, the molecular machinery that we'll talk about later, the repair machinery, will come in and it will fill in these gaps that we see that were created, the eroded strands. And so we'll get what's called this heteroduplex region where these 4 strands that were independent have now crossed over and connected to each other, and we can see that no longer is this chromosome here no longer is completely red. It's crossed over with one of the blue chromosomes, and actually all 4 of them have exchanged parts of their chromosomes for one another. And that's kind of the onus of crossing over, is we've now mismatched the genes between 2 different chromosomes. And so this kind of disrupts that linkage that we talked about earlier. So that's all for this chapter. We'll continue on with chapter 5.